Hey kids, it's Mrs. Frazel. How are you? I hope you're fine. I apologize for not having this up sooner. I should have had this up for you guys to do when I assigned it, but that's okay. So if you are struggling with figuring out how to run the lab on this protein synthesis mutations simulation, here you go. I'm just going to run through it really quickly with you. So when you click this link that's on the worksheet, it will take you to this website. Okay, so this is a really awesome way to visualize how protein synthesis is done and what mutations do to protein synthesis, but the actual page itself is a little bit tricky to run. So here you go. This green box right here is where you're going to start, and what this is this is showing you the sequence of nucleotides, the A, C, T's, and G's, in the gene that codes for an enzyme called lactase, which is how we digest cow's milk protein. So what you're going to do first is just practice running protein synthesis on that gene. So the first thing you have to do is push setup, and that is going to show you the DNA strand for the lactase gene-ish. Then what you need to do is click Go Stop. You need to make sure that this Go Stop button is dark blue. Okay, and then you'll just go through 1, 2, 3, and 4, and you need to take your time because the simulation takes a minute to do each of those things. So give it its time to go through each animation, okay? So there's transcription, made the mRNA, you released it, then you translate it. And translation makes the amino acid sequence or polypeptide appear. So you can see the names of the amino acids here, okay? And then releasing it just makes it easier to see because you get rid of the tRNA, okay? Easy peasy, right? And you'll answer the questions just describing what's happening as you're going through that. Okay, and then the second part, in the second green box, you're going to take a look at how a mutation will change it. So you're going to leave this here, come down to this second box, okay? This first green box, you can change the number of nucleotides that you change in this original DNA strand, okay? So this sequence down here in the cell B DNA, that is the same sequence as up here, but with one change somewhere, okay? And this has made a substitution change, but if you look back on your worksheet, create one deletion mutation. Here's how you do that. Click here on mutation type, drop down, deletion instead of substitution. And what you need to do then is click 5. So it's going to put in the DNA strand again, only it has deleted one nucleotide. And you're going to look at how it affects that. So then run through the transcription again. Oh, look at that. What's different there? Release it. Translate it. Release it. You don't need to worry about the show protein production. But you want to then compare this amino acid sequence with the original amino acid sequence that it should have made. Okay? And answer the questions about that. Okay, so that goes through here. Now, if we take a look at this protein, it's significantly shorter, right? It shifted the reading frame on the codons when we did that deletion. So it has changed the order of the amino acids up to where that deletion occurred. Okay? So it changed right about here. So we have the phenylalanine and then isoleucine there, and then alanine is next. That's right. But when we get to the next one, instead of coding for leucine like it was supposed to, it coded for cysteine, and then it just didn't know what to do from there. Okay, it had a stop codon. Okay, So there was your practice run. Now for the second investigation, you are going to do that same thing only on the DMD gene. And what you need to do to fill in this table is first read the instructions, because you always read all of the things, but in the green box, you're going to set the gene to the DMD gene. So that is this one up here. Okay, so we're going to reset it from lactase to DMD and then click setup again. That's going to erase everything that you just did, reset the gene to a different sequence, which is to mimic the DMD gene, and then you'll run all of that again. Okay, so there's the amino acid sequence that you should have in order to have a functioning strand of which protein does the DMD gene make? Yeah, dystrophin, you're right. So what you're going to do then is come down here and you're just going to choose some random mutations. What do you want to do to this gene to take a look at how the dystrophin protein responds? Okay, so again, let's do one deletion and run it from there. Okay, and see what happens. Oh, look at that. That's really short. Okay. So we hit another stop codon too soon. Okay, so when we look at this one, it stops after the second glutamine. Okay? That protein is significantly shorter than the original protein. It is probably not going to function, right? Because proteins have to have a specific shape and size in order to do their job. And if you remember the models that we looked at for how dystrophin functions in the muscle, is this tiny little protein going to be able to reach from the actin to the sarcolemma to help do its job? Okay, that's what you want to look at. What you're going to do is, as you fill in this table, just choose a different type and number of mutations. So that was, the first one was a deletion. We changed one. We're going to count the number of changed amino acids in the protein. So we have, I believe there's 19 amino acids in this strand in the original, if you count them. So 19, we had 1, 2, 3, 4, 5, 6 in the mutated strand. So 19 minus 6. But Frable's not a math teacher, but I'm pretty sure that that is 6. So that's only 13 changes. And probably, I mean, you're going to do your best to kind of figure this out on your own. If the rule of thumb kind of is, if it's more than a 10% change in the number of amino acids, it's probably not going to be a functional protein. If it's a few changes, it might still function. It will probably still function. Okay, so just go through, choose the different levels and different numbers of mutations you're going to do, and then rerun it every time and see how it goes as you fill in that table. Okay. Okay. And then make sure that you answer that question. Then you should make sure you read through all of the different types of mutations and answer these questions as they applied back to what you looked at the lab. Okay. Okay. If you have any questions, if this wasn't helpful, for sure send me an email, and have a great day. Bye-bye.